Today we talk about endocrine system, and this lecture includes both central and peripheral endocrine organs. So please be attentive, and the lecture is very concentrated. So if you are tired, then take a rest, drink a coffee, and after that continue to listen, because the topic is very important for every doctor. Okay, first about endocrine organs. You remember that these glands, they inject their hormones to the bloodstream or to the lymph as they are ductless, comparing with exocrine glands. And endocrine organs, they are divided into the central and peripheral. And usually students say, when we ask them why some organs are considered as central, they say because they are located in the central nervous system. That's not true, actually. And the correct answer is that central organs, they control the activity of peripheral. And those central organs, they include hypothalamus, hypophysis or pituitary gland, and epiphysis or pineal gland. And let's start our lecture exactly from them. Hypothalamus. From anatomy you know a lot about this structure, and today we consider it not as a part of the nervous system, but as a part of endocrine system. As some nuclei inside it, they produce not only neuromediators, but hormones as well. And those hormones could be divided into two broad groups. First group of hormones, they affect smooth muscle cells in the peripheral organs. And those two hormones, these are oxytocin and vasopressin. What do you have to remember? The name of the nuclei producing them and the effect of each hormone. So, paraventricular nuclei of hypothalamus, they produce oxytocin. This hormone causes the contraction of smooth muscle cells in the uterus during the birth, for example. Also, they affect smooth muscle cells in the mammary gland during lactation. And oxytocin is widely used to stimulate the delivery in case the natural production of oxytocin is low. And paraventricular nuclei are called so because they are located by each side of the third ventricle in the hypothalamus. Then, the second group of nuclei, the second pair, is represented by supraoptic nuclei. They are called so because they are located above the chiasma opticus, which is attached to the brain at its base. Here is the chiasma opticus, here is the brain, and exactly here above the carotid artery there is supraoptic nucleus. So it produces predominantly vasopressin, and another name of the hormone is antidiuretic. You should remember both as they are equally used. From the name we guess the function, as vasopressin means vessel compression, and they stimulate the contraction of smooth muscles inside vessels, and in this way they increase the blood pressure. Another mechanism of their action is through the kidneys. They decrease the diuresis. That's why another name is antidiuretic. And decreasing the diuresis, they keep more water in the organism, inside the bloodstream. And if there is more water inside the vessel, then the pressure is elevated. So vasopressin or antidiuretic hormone, they increase the blood pressure via two different mechanisms. And deficiency of this hormone stimulates the diabetes insipidus, which is accompanied by the active urination. Okay, then another group of hormones produced by the hypothalamus is destined to control the activity of pituitary gland of hypophysis. And that's why hypothalamus is considered as the central endocrine organ, as its hormones, they control the anterior pituitary. And let's consider these hormones, let's return to them after we investigate the pituitary gland, because before understanding how this control is realized, we should understand what cells are controlled by these hormones. And as for the specimen, once again, as we have just considered, in the specimen you should recognize the third ventricle cavity and the chiasma opticus. These are the two key structures, and with the help of which you can distinguish respective nuclei. These are the paraventricular like butterfly wings. They are found on each side of the third ventricle. And here are the supraoptic nuclei above the chiasma opticus. Not to confuse with the suprachiasmatic nucleus, okay? Then, after that, we consider the pituitary gland, which is connected tightly with the hypothalamus, both structurally and functionally. So first we consider the structure of the pituitary gland, and after that we discuss the interrelationships between the hypothalamus and pituitary. So first, adenohypophysis is also called anterior pituitary, and it is represented by three parts. The most important and the largest one, the bulk of adenohypophysis, is made up of pars distalis. Here it is, the largest one. And two other, these are the pars intermedia between the adenohypophysis and neurohypophysis. Here it is. And the pars tuberalis. Pars tuberalis, here it is. Pars tuberalis is it surrounds the infundibulum of the neurohypophysis. What about the posterior pituitary, neurohypophysis? It is represented by two parts, pars neurosa, the largest one, and the infundibulum. Here it is. Notice that infundibulum connects hypothalamus with the pars neurosa. So neurohypophysis is structurally connected to the hypothalamus, and actually from histological point of view, neurohypophysis is the continuation of the hypothalamus. Okay, and now let's investigate it in details. Neurohypophysis is represented completely by axons descending from the hypothalamus. So what is interesting is that neurohypophysis has its own neurons. It contains only axons of hypothalamus. And those axons, they are dilated end portions destined to store, to sequester hypothalamic hormones. So we have just discussed the hypothalamus with its hormones, and now we should understand that hormones are only produced there, but after that they descend to the neurohypophysis, to the posterior pituitary, and are sequestered there. So, neurohypophysis is the storage for hypothalamic hormones. Okay. And now, what is another important thing is that pars neurosa, it sequesters oxytocin and vasopressin. Okay. While in the infundibulum, especially in the median eminence, we can find the storage of another hypothalamic hormones. Those hormones, they control the activity of adenohypophysis. We'll consider them later, okay? So as I told you, in the hypothalamus, two groups of hormones are produced, oxytocin and vasopressin, and hormones controlling the adenohypophysis. So the first group is sequestered in the pars neurosa, another group in the median eminence, and that's what we are to discuss later. Now, what term you should remember? This term is Herring bodies. The definition is that Herring bodies, they represent the dilated end portions of axons with oxytocin and vasopressin inside. So usually neurons and their axons, they terminate on another neuron to conduct the nerve impulse to another cell or to the target organ. But here, dilated portions of axons, they end on the blood capillaries to inject their hormones to the bloodstream. So Herring bodies, these are the storage. These are the end portions of axons, and they are destined to sequester hormones and to inject them into the bloodstream when it is needed. Okay, and now what we should highlight, as here we can see the intimate interrelationship between the neuron and the blood vessel. So guess what is absent between them? Which cells are usually present between the neuron and the blood vessel? And those cells are absent here. And if you study well, you remember that this is the exactly blood-brain barrier made up of astroglial cells. So usually astrocytes are present between the vessel and the neuron, but here in the neurohypophysis, this barrier is absent. That's why neurohypophysis belongs to the so-called circumventricular organs. In these organs, barrier is absent because neurons, they produce hormones, and hormones should be injected to the bloodstream. So blood vessels should be of increased permeability. Here we have fenestrated capillaries, and blood-brain barrier is absent, so these organs are more threatened by infection comparing with other parts of the central nervous system. Okay, so after we have discussed the neurohypophysis, and now we comprehend that actually it has its own neurons, and this is just the storage of hypothalamic hormones. Now we go to the anterior pituitary and investigate cells inside this part of the organ. So cells of adenohypophysis, they are classified based on their staining properties. Some cells are considered as chromophobic, as they are labeled with the letter C, and they are neutral. They don't like to be stained, so they are afraid of colors from their name. And these are either undifferentiated cells or maybe cells just after they have discharged their hormones to the bloodstream. So the function is not very well understood, but 50% of cells are chromophobic. After that, chromophilic cells. Depending on the dye they love, either acidophilic or basophilic, they are classified into acidophils and basophils. Acidophils, they have a little bit acidophilic granules inside, here they are, and basophils, basophilic granules respectively. And that's what you have to remember, because this is the basic information for the minimal mark. Acidophilic cells are classified into somatotropes and lactotropes. Somatotropes, they produce growth hormone. Lactotropes, they produce prolactin. And first of all, I want to highlight that somatotropic hormone or growth hormone and prolactin, they affect directly the target organ. And that's why it is better not to call them tropic, because tropic hormones means that this hormone affects another endocrine gland. In case of growth hormone and prolactin, such hormones, they target exactly the peripheral organs, not another endocrine glands. That's why cells are called somatotropes and lactotropes, but hormone, it is better to say growth hormone and prolactin. Okay, so it controls the growth and lactation of the mammary gland respectively. After that, as for the basophils, they are classified into three groups. Gonadotropes, producing luteinizing and follicular stimulating hormone in the gonads, ovaries or testes. Then thyrotropic cells, they produce thyrotropic hormone, activating follicular cells of our thyroid. And at last, corticotropic cells, they produce corticotropic hormone, regulating the activity of the zona fasciculata in our adrenal cortex. At the end of the lecture, in the second part, we consider the structure of thyroid and adrenal, and it will be much more clear for you. So you may then return back here to understand better the function and targets. But what do I want to highlight? First of all, corticotropic cells, they produce corticotropic hormone in form of large precursor, which is called pro-opiomelanocortin. Pro-opiomelanocortin, and this large polypeptide is then cut into smaller one, and a piece of this large peptide is corticotropic hormone. But another piece is melanocyte stimulating hormone, and it causes changes in pigmentation. Why is it important to understand? Because in case of hyperfunction of these cells, not only the level of corticotropic hormone elevates, but also melanocyte stimulating hormone, and it causes changes in the skin color, and this is a very important diagnostic criterion in endocrinology. That's why you should know about it. I didn't put it in text, but please read in the textbook about the pro-opiomelanocortin. Has very high significance, practical significance. As for the gonadotropic cells, notice that each cell produces its own hormone, but gonadotropic cells, they produce two, follicular stimulating or luteinizing. In details about the effects we talk in male and female reproductive systems. So today I don't want, you shouldn't remember the effects. They will be discussed later in another topics. Okay, so now we understand that there are 5 types of cells producing 6 hormones, and the question is how the activity of those cells is regulated. To understand that, let's return to the hypothalamus and to the second group of its hormones. This group of hormones controls the activity of adenohypophysis of those 5 types of cells with the help of 2 groups, releasing hormones or liberins. They stimulate adenohypophysis to produce something, or statins. Those hormones, they inhibit the activity of anterior pituitary. And now how to remember all those hormones? Very easy to remember. If you remember 5 cells in the adenohypophysis, somatotropes, lactotropes, thyrotropes, gonadotropes, and corticotropes, each cell has its releasing hormone except lactotropes. So 4 of them, they have their own releasing hormone: thyrotropin releasing hormone, gonadotropin releasing hormone, growth hormone releasing hormone, and corticotropin releasing hormone, except prolactin, because prolactin is stimulated by the thyrotropin releasing hormone. Okay, and prolactin, it has its own statin. So prolactin could be inhibited with the help of dopamine. Dopamine is the statin for prolactin. And another only one statin you should keep in mind. This is the somatostatin. And this hormone you remember already from the topic pancreas, as it was produced by the islets of Langerhans, and it inhibits the activity of endocrine and exocrine cells of the pancreas. And now this hormone produced in the hypothalamus, it also inhibits the growth hormone. So somatostatin is the statin for the somatotropes. It inhibits the activity of the growth hormone. Okay, so let's move on. And now our purpose, besides, in the anterior pituitary we also can find a special subtype as follicular stellate cells. They are not very important because their function is not completely understood. It was supposed that they are the stem cells, but it is not evidenced up to now. But just remember, they form the small follicles, and they are connected with the gap junctions, and they might cause the regulatory action on the cells on the of the anterior pituitary. Then, what about the hormones? Now, after we have considered the hypothalamus with its releasing hormones and statins and pituitary gland, we have to say that there is a special mechanism controlling the production of hormones. So the question is why and how hypothalamus know whether to produce liberin or statin, how to balance the concentration of the target hormone. The answer is in the negative feedback. And negative feedback mechanism means that the target hormone, the hormones of thyroid gland, they have the negative feedback effect on the production of the thyrotropic and thyrotropin releasing hormone. So once hypothalamus produces its thyrotropin releasing hormone, it stimulates pituitary to produce thyrotropic hormone. Then thyrotropic hormone stimulates thyroid to produce T3 and T4. And what happens after that? Negative feedback. T3 and T4 inhibit both pituitary and hypothalamus, informing that these hormones are already released, so stop stimulating their production. Okay, then now let's summarize the information about the hypothalamus and hypophysis and integrate them in a single system, hypothalamo-hypophyseal system, as they are really connected structurally and functionally. So the structural link is that neurohypophysis is the structural continuation of the hypothalamus, as it contains axons either with oxytocin and vasopressin in the pars neurosum, or axons with liberins and statins, sequestered inside. So the neurohypophysis is the structural continuation of the hypothalamus. Then, what about adenohypophysis? How does it relate it with hypothalamus? Adenohypophysis is under the control of hypothalamic hormones, liberins or statins. So if liberin is produced, then adenohypophysis produces its hormone. If statin is produced, then it stops its production. And the question is, how do those hormones from the hypothalamus, from the nervous system, are delivered to the adenohypophysis, to the endocrine system? And the answer is in the blood supply, hypophyseal portal system. Very easy to comprehend if you understand the destination of those vessels. So let's consider together. First of all, in the hypophysis, superior hypophyseal artery brings the arterial oxygenated blood to the median eminence. Here it gives rise to the primary capillary network, which are fenestrated, so of high permeability. Why they are fenestrated? Because here not only exchange of gases takes place, but also the liberins and statins are absorbed to the blood through fenestrated capillaries. So the purpose of this capillary network is not only to bring oxygen, but also to take hormones. That's why they are fenestrated, that's why blood-brain barrier is absent. Okay, once they absorb them, they deliver those hormones through the hypophyseal portal venules, here they are, from the median eminence, from the nervous system, to the adenohypophysis, to the endocrine system. And here, those venules, they dissipate in the secondary capillary network, which is completely venous. And as you can see, those capillaries are blue. They do not contain oxygen. So those capillaries are destined not to exchange gases, but to exchange hormones. And they are also fenestrated type. And here we see that liberins and statins, they are discharged. They cause their effect on adenohypophysis, and hormones of adenohypophysis are absorbed to those capillaries and then distributed through all our body and affect target organs. So the secondary network is destined to bring liberins and statins and to take respective hormones of adenohypophysis. Exchange of hormones takes place, not gases. That's why pituitary gland is considered as an unusual organ, because it has capillary network without destined not to exchange gases. Okay, I think now we have considered all the most important aspects, and you understand that really hypothalamus and hypophysis, these are the single system, and we always should consider them together because they are related structurally and functionally. Yeah, besides, I forgot to say about the adenohypophysis pars intermedia and pars tuberalis. What about their function? They do not have their distant important functions. In the pars intermedia, in fetal period of life, melanocyte-stimulating hormone is produced. That's why in pregnant women, skin changes the color, especially areolar part of mammary gland, because in pregnant women, melanocyte-stimulating hormone from the fetus circulates in the blood. But in adults, this hormone is not produced, and it seems the function is rudimental. Also, some follicles could be found there, but that's not very important. Pars distalis has the most important function. Yeah, and in the pars distalis, after the pregnancy, the portion of lactotropes proportion increases. That's why pituitary gland of women after the delivery is much larger than the pituitary gland of all others. Then, embryonic development. Very important to say that neurohypophysis is formed like outgrowth of the hypothalamus, floor of the third ventricle, a part of diencephalon, and it keeps this connection through all the life. While adenohypophysis, it derives from the oral cavity ectoderm, please ectoderm of the oral cavity, and it is also called Radke's pouch, and it loses its connection with the oral cavity, migrates upwards, and eventually it is enclosed with the same cella turcica together with the neurohypophysis. And here they are in very close proximity, and they are considered as the single organ, despite they have two different functions, structure and embryonic development. Why is it important to understand? Because sometimes in the oral cavity some remnants of pituitary gland might be found, and because some surgical intervention, it is more easy to perform through oral cavity than to reach the hypophysis through the large hemispheres. Okay, and as for the specimen, what should you recognize in the specimen? This is the neurohypophysis and this is the adenohypophysis. Notice the difference is in the staining properties. Neurohypophysis is more palely stained, while adenohypophysis is very intensely. Notice that neurohypophysis in this case is put in front of adenohypophysis, despite it is considered as posterior pituitary, because the specimen could be, reflected, put on either side, so it doesn't matter, it is in front of adenohypophysis or behind, because you should distinguish them based on the structure, not based on their position. That's what I want to say. So do not confuse them. Neurohypophysis is more palely stained, and there are no neurons, only nerve endings, and those cells we can see there, these are astrocytes, these are glial cells, and they are called pituicytes, and they do not perform any important, just distant function. Then, what about the adenohypophysis? This is the pars distalis, the largest one. Between the pars neurosa and the pars distalis, there is the pars intermedia with small follicles inside. And around the infundibulum, this is the connecting stalk, and it connects the neurohypophysis to the hypothalamus. Around the infundibulum, we can find the pars tuberalis. Okay, that's what you have to distinguish. Notice that some cavity could be seen inside the neurohypophysis, and this is the continuation of the third ventricle, and this cavity is the remnant of the Radke's pouch, as Radke's pouch is a vesicle, and inside it there is a cavity, and this cavity remains in adenohypophysis. Okay, now the third central gland is pineal, and it is hidden under the large hemispheres, and in humans it is a little bit rudimental, and it is formed in embryonic development as the outpocketing of the roof of the third ventricle. So if pituitary gland is formed at the base, then pineal gland is formed from the apex, from the roof of the same cavity. Then, what about the functions of the pineal gland? It controls the photoperiodicity, or so-called circadian rhythm, or 24-hour rhythm. You know that along the 24 hours, we have the period of sleep and the period of activity, and the alternation of both depends on the level of the light. So usually at night we are less active, we sleep, at day we are more active and we do what we usually do. And the organism, it changes the activity depending on the level of light, which is converted into the level of two respective hormones. So at night, pineal gland, with the help of pinealocytes, produces melatonin, which boosts our imagination and at the same time decreases the activity of the majority of organs. And at day, it produces serotonin. Serotonin, this is the hormone of the high spirit, good spirit, and the hormone of happiness, in other words. So at day we are more active due to serotonin, and the combination of those two hormones help us to inform when to go to bed, when to wake up. And you know the problem with those two hormones: when you have a long flight, when you change the longitude, when you travel for very long distances, when the time zone is changed, so you have some time to adapt. That's because your hormones are produced in the rhythm characteristic to the previous place of location, and you have some time to change those photoperiods. And that's why pineal gland is considered as the central, as it controls the activity of all the other organs, depending on the period of night and day. As for the embryonic development, I've said, and with aging, it accumulates their calcium crystals inside, and they make it like the pine cone. That's why pineal gland is called pineal. Looks like pine cone. And those crystals, they are very prominent even in the X-ray, and that's why this gland is used to distinguish the central axis of the brain, because it is very prominent. These crystals are called corpora arenacea, or brain sand, and this is the normal event when this sand appears in the pineal gland. And what I forgot to say is that except pinealocytes producing those two hormones, astroglial cells are present as well, because this is the part of the nervous system. Yeah, besides, notice that blood-brain barrier is also absent here. What about its function, how it works, how it detects the light in the outer environment, and how pineal gland understands what hormone to produce? In primitive animals, pineal gland is formed like a third eye, and maybe it is not very well seen. Let's put it here. Okay, and you see this lizard, and part of the occipital bone is transparent, like a cartilage, and through this transparent cartilage, semi-transparent light directly affects the epiphysis. And also they are informed about the danger, and some motion maybe behind them. In humans it doesn't work. Why? Because the third eye is no longer needed, because our pineal gland receives the information from the brain, or from our retina. So in our eyes, there is a special retina hypothalamic tract which deliver the information to the pineal gland. There is the word put especially for me, the K word is melanopsin. You remember in our rods and cones there were two pigments, rhodopsin and iodopsin, and they are responsible for the visual information. But also there is one more pigment, visual pigment is melanopsin. This protein is expressed by ganglion cells, and it is not responsible for the visual information. It doesn't give us the image, but its function is just to inform us about the light. So if our retina is enlightened, then melanopsin sends the information via this tract to the pineal gland to produce which hormone? Serotonin. And serotonin is converted from the tryptophan amino acid. That's why this amino acid should be supplied normally to stimulate the normal production and level of serotonin. So melanopsin is a special opsin responsible for the information responsible for detection the light, and this information is exactly used by pineal gland to produce serotonin. Then serotonin makes us happy, and this hormone is very important for all who wants to be a psychologist. If you want to work in field of psychology, psychotherapy, psychiatry, then it is very important to understand the mechanism of its production. And I want to say that the majority of serotonin is produced by our intestines after we have eaten something. But in our intestines it is used like neuromediator, but also as the hormone of happiness. And okay, that's who is interested, you will read more about this hormone. Then, what about the melatonin? Melatonin is produced at night. It is responsible for the imagination. That's why we are afraid of something more at night than at day, because of melatonin particularly. And what is more important, as for you as future doctors, that melatonin, it regulates reproductive function, inhibiting the steroidogenic activity, inhibiting it. So melatonin and pineal gland in humans, it prevents early onset sexual maturation. So in children, pineal gland prevents preliminary sexual maturation. So in case pineal gland is destroyed, this sexual maturation might occur before the term, before the puberty. And in animals, this gland, pineal gland, it controls the periods of reproduction. You know that animals, for example, they reproduce in spring predominantly. Why? Because the ratio between the day and night changes depending on the season. For example, now we have night much longer than the day, so more melatonin is produced, less serotonin, and that's the information helping to control another system of organs. And in animals, melatonin is produced in high amount. It inhibits the sexual activity, so it prevents the reproduction during this season. Okay, so the epiphysis is a biological clock helping us not only to detect the period of a day, but also to detect the period of a season. And very important information is that seasonal affective disorders are based on the activity of pineal gland. Now, when we have very long night, very short day, less serotonin is produced. That's why some people, they are suffered from the seasonal affective disorders, and they are treated with serotonin and with many drugs. In pharmacology, you will investigate how to manipulate the concentration of serotonin. Okay, very interesting. If you are interested, you'll read more about this, but we have to go further and to investigate thyroid, parathyroid, and adrenal glands now. What about thyroid? This is the peripheral gland. It is under the control of thyrotropic hormone and thyrotropin releasing hormone of the hypothalamus, and it is represented by a system of follicles, spherical vesicles, with a special viscous substance colloid inside them. So here this follicle is shown. It is made up of one layer of epithelial cells called follicular cells, and this viscous colloid contains a special protein called thyroglobulin inside. And first of all, let's remember that there are two types of cells in the thyroid gland. Very important. They have different origin, different function. So follicular cells, they represent the majority of cells, and they produce two hormones, T3, which is much more active, and T4. Together, they increase the metabolic rate. They stimulate the oxidation processes, so more ATP is produced, and about the effects we'll talk about later. And C cells, they are less numerous. They do not reach the lumen of the follicle. They are located close to the basement membrane here, and they contain the granules with calcitonin. Calcitonin is the hormone helping to decrease the blood calcium, and C cells, they produce calcitonin in case the blood calcium is elevated. So they are not controlled by another hormones, they are directly regulated by the calcium level. So remember, C cells, they decrease the blood calcium with the help of calcitonin. Okay, let's move on. And let's consider the steps of T3 and T4 production, as it is very important, and those hormones, they have the very high clinical significance as they affect all systems of our body. In endocrinology, you will investigate patients with the hyperfunction and the hypofunction of the thyroid gland. And they differ significantly because people with high hyperfunction of thyroid gland, they talk too fast, they do everything very quickly, they are very active, they can't concentrate their attention, and they are prone to lean, so they have low body weight. And people with a hyperfunction, as their metabolism is not stimulated, they are prone to obesity, they are very slowly speaking, they do everything very slowly, and that difference is very obvious in their behavior. So once you see them, you will remember forever. And now, what about the formation of those hormones, how this mechanism is controlled? It is very complicated, but each step could be broken. So to understand what is the correct diagnosis, or what is the point of mistake, you should understand the complete mechanism. Let's start. from the beginning. So these are the follicular cells of our thyroid. And the first step is to produce the thyroglobulin on the rough endoplasmic reticulum. So these cells, they have very well developed rough ER. It produces thyroglobulin, and through Golgi it exocytoses it to the colloid. Notice that thyroglobulin is a polypeptide, is a protein, and it is rich in the amino acid tyrosine. Tyrosine is the precursor for the T3 and T4. Okay. So thyroglobulin is not a hormone, just a lone peptide with many tyrosine residues in it. Then iodide, which is a very important component of the T3 and T4, iodide is absorbed through the base of the cell with the help of the sodium iodide symporters. So together with the positively charged sodium, iodide enters the cell, then passing through it, it is delivered to the colloid through a special exchanger called pendrine. It exchanges iodide with a chloride. So chloride enters the cell, iodide is expelled out of the cell to the colloid. So iodide passes through the cell with the help of two proteins. Then the next step is controlled by the thyroid peroxidase. It oxidizes iodide and stimulates iodination of tyrosine inside thyroglobulin. So thyroid peroxidase, it performs the very important function, the iodination of tyrosine residues. And sometimes during the autoimmune reactions, thyroid peroxidase is rejected by the immunity, so autoantibodies are formed. That's why you have to know about these diseases, about this protein. So everything has its clinical significance. So inside the thyroglobulin, tyrosine residues could be iodinated with 2 or with 1 iodide. So T2 or T1 is formed. So tyrosine residue could be iodinated 2 times or only 1 time. Then 2 neighboring tyrosine residues, they are coupled together. And in case T2 and T1 is coupled, T3 is formed. In case T2 are coupled, T4 is formed. So the hormone of thyroid, T3 and T4, made up of 2 tyrosine residues, each of which could be iodinated 2 times or only 1. Okay, then, so in this form, T3 and T4, they are sequestered inside the colloid, so they are not active because on this step, T3 and T4, they are incorporated in the polypeptide chain thyroglobulin, so they are not active. And thyroid is an unusual gland because it sequesters its hormones outside the cell. We can find granules with hormones inside, but instead we see inactive hormone in the colloid. After we need a hormone, T3 and T4, pituitary gland produces thyrotropic hormone. Thyrotropic hormone stimulates follicular cells to endocytose, a part of thyroglobulin. So endocytotic vesicles are formed. And this is the mechanism of receptor-mediated endocytosis. You remember from cytology. Okay, so when thyrotropic hormone informs follicular cell that we need T3 and T4, this cell decides to endocytose a part of thyroglobulin, and then 2 pathways exist. The predominant pathway is lysosomal. When endosome fuses with a lysosome, and as a result, lysosomal enzymes, they cut thyroglobulin into amino acids, and they cut off this T3 or T4, and as a result, they are injected to the bloodstream. This is the predominant pathway. So once we need a hormone, thyroglobulin is endocytosed, broken down with lysosomal enzymes into amino acids, and as a result, T3 and T4, they are injected to the bloodstream. But another way is transepithelial. When thyroglobulin just after it is endocytosed, it is exocytosed as a thyroglobulin outside the cell. Okay, unchanged form of thyroglobulin. And this is mediated through the megalin receptor. And this pathway is used in case the follicular cell is overstimulated, just not to produce too much T3 and T4. Okay, so you should know the difference. In case of lysosomal pathway, thyroglobulin is cut into amino acids. In case of transepithelial pathway, thyroglobulin is exocytosed in unchanged form. As a result, eventually T3 and T4, they appear in the bloodstream. What happens further? In the blood plasma, less than 1% is transported in unbound form, transported as free hormones, and they're used for the negative feedback mechanism. But predominantly, hormones T3 and T4 are bound to the thyroxine-binding globulin. This is the one transport form. Another transport form is transthyretin. It is called so because it transports not only thyroid hormones, but retinol as well. So it is produced in the retina, it is found in the cerebrospinal fluid, doesn't matter, just this is the second transport protein. And some other protein like albumins, they also could transport thyroid hormones. What is important is that hormones, those transport proteins could be measured in the bloodstream, and this is the important parameter. So you should know they have the clinical significance. Then, once hormones T3 and T4 arrive to the target organ, formerly it was thought they just simply diffuse through the membrane, for example, through the membrane of the neuron. But now we know that there is a special transporter proteins helping T3 and T4 to pass across the membrane of the target cell. Why is it important information? Because in case those receptors, transporters, are mutated, some organs might be insensitive, might lose sensitivity for the thyroid hormones. So even in case thyroid hormones are normally produced and transported, sometimes some organs, they do not get any effects from them because their transporter proteins are mutated. For example, those two are used in the brain. So I put them here. These transporters are used also to transport some other molecules, but they are also important to transport T3 and T4 inside the cell. Okay, next step is that thyroid hormones, they interact with their receptors, and those receptors, they are transcription factors because they affect the transcription of some genes in the nucleus. So they change the transcription program, the behavior of the cell, they increase the metabolic rate, they stimulate cell to burn more glucose, to produce more ATP, and that's what increases the heartbeat rate, increases a little bit the blood pressure and all the reactions. And in case it doesn't occur, blood pressure will be lower and less glucose will be oxidized, so more glucose will be sequestered and so on. Then, what about the conversion of T3 to T4? As I told you, T3 is more active. That's why in tissues, special deiodinases are present to convert T4 into T3. And also special deiodinases are used to inactivate both. So inside tissues, those special enzymes are present, and you should know about their function. As you can see, lots of information about thyroid gland, but each protein put here has high clinical significance. That's why we represent this information. So in case you want to get 5, you should know not only about types of cells and hormones, but also the steps of its synthesis and action. It will help you a lot in endocrinology department, I promise you. So, the development of thyroid gland, it has 2 sources of development. Both are located in the pharynx, in the endoderm, but thyroid gland itself and its follicular cells, they derive from the oral cavity, from the endoderm, between the first and second pharyngeal pouch, like invagination of the endoderm downwards, and a special thyroglossal duct is present and it obliterates under normal circumstances, and sometimes it forms the isthmus of the thyroid gland, as it has two lobes and they are connected by the isthmus, which is formed out of this duct. Sometimes in the foramen secum of the tongue, some remnants of the thyroid gland might be left. So that's why we investigate the embryonic development. So we should understand and keep in mind that thyroid gland migrates from the oral cavity, from the pharynx if to be exact. Then what about the C cells? C cells, they derive from the endoderm of the fourth pharyngeal pouch, and also endoderm, but of a little bit another region. And formerly it was thought that originally they immigrate from the neural crest, and later that's not true. So if you find anywhere a neural crest, then forget about it. Seems that they derive from the endoderm of the fourth pharyngeal pouch. Okay. As for the specimen, that's what you should recognize. Very difficult to confuse thyroid with any other gland, as it has very promiscuous structure. Here follicles can be distinguished. They are formed by a single layer of epithelial cells, and inside colloid we can find those vesicles, which are the signs of endocytosis of a portion of colloid. So once follicular cell eat a piece of colloid, this empty space is formed. Okay, besides, notice that C cells cannot be distinguished with a light microscopy, and fenestrated capillaries are found here everywhere, and rich capillary network surrounds each follicle to absorb hormones readily. Then parathyroid gland, there are 4 of them incorporated inside the thyroid, and we say that there is an upper pair and a lower pair. Collectively there are 4. And in histological structure we distinguish the oxyphil cells. Here they are a little bit more oxyphilic, and principal cells, which are more basophilic, and they produce parathyroid hormone. Parathyroid hormone, it elevates the blood calcium, and very easy to remember, because C cells they produce calcitonin to decrease the blood calcium, while principal cells of parathyroid gland, they produce parathyroid hormone to elevate the blood calcium, and together they balance the blood calcium. So these cells, they are controlled by the level of blood calcium. In case it decreases, principal cells, they produce more parathyroid hormone to stimulate the calcium resorption, absorption, and reabsorption. So the effects of parathyroid hormone is better explained in our topic bones, and return back to that lecture, and about osteoclast, particularly this chapter, because they realize the effects through osteoclast, and they affect osteoclasts not directly, but this hormone acts through the rancle and osteoprogerin system. So if you are interested, you return back and investigate this mechanism in details. Now, in general, you should keep in mind that parathyroid hormone not only stimulates the bone resorption, so it doesn't only take calcium out of there, but also it stimulates reabsorption of calcium from kidneys, and absorption of calcium in the intestine from the food. Notice it also stimulates the vitamin D hydroxylation in the kidney, because this vitamin participates in the calcium metabolism as well. So it has the complex effect, and if you're going to work in traumatology, it's very important to investigate in detail some. As for the embryonic development, again we talk about the pharynx. We have just said that thyroid gland is formed from the bottom of the pharynx, and C cells, they are formed from the fourth pharyngeal pouch. And now we should remember that parathyroid gland is formed from the third and fourth pharyngeal pouches, and upper pair derives from the fourth pouch, and lower pair derives from the third. So the third pouch, they migrate lower and transform into lower part. lower pair of parathyroid glands. Notice that third pouch is also used as a source of thymus. So some defects of the thymus and some immunodeficiency syndromes, they are accompanied with the problems of calcium level, calcium metabolism. And this migration usually is due to problems with the neural crest. That's why abnormalities with the thymus, parathyroid gland, they are usually accompanied with problems with our ears and heart defects. That's why it is very important to check all another abnormalities in case of one mutation, one defect is detected. So in case we understand that there is the problem with the thymus, we should check the parathyroid glands, and heart defects and many other congenital defects should be also checked. Okay, let's move on. And the last gland we are to consider today, this is the adrenal gland. And it is represented by the cortex and the medulla. Cortex is divided into 3 zones, each producing its own group of hormones, each having its own regulation. And the medulla, which has quite another embryonic development and function. One by one. So here the concentrated information is given. This is the basic information you have to remember. So cortex, covered with a capsule, is represented by 3 zones. They are usually named in Latin: zona glomerulosa, zona fasciculata, and the zona reticularis. Glomerulosa, because it is made up of glomerulus, ball-like structures, cellular aggregations, and they produce mineralocorticoids. From its name, corticoids highlights it is produced by the cortex. Minerals means it regulates the ion balance. It regulates the sodium and water metabolism. In detail, the most important representative is the aldosterone. And aldosterone production is controlled by kidneys, by a special renin-angiotensin-aldosterone system. This system is considered in details in the lecture about kidneys, so we'll discuss it later. Now just remember that mineralocorticoids and aldosterone regulate the sodium and water metabolism, and together with that the blood pressure, because the water and sodium balance, if in case there is more water in the bloodstream, then the blood pressure is elevated. So regulating the amount of water in our vessels, we regulate the blood pressure in this way. Okay, so this is, as for the zona glomerulosa, it is under the control of kidneys, under the renin-angiotensin system. Then the thickest one, zona fasciculata, it produces glucocorticoids. Corticoids means they are under the action of, they are produced by the cortex. Glucose means it controls the glucose metabolism. In general, it stimulates gluconeogenesis, formation of glucose from another chemicals like carbohydrates, like nucleic acids, amino acids, and so on. So formation of glucose from another molecules. But the most important action is to inhibit the inflammatory reactions. Besides, the most well-known hormone is cortisol. And you remember, maybe you've heard already, that in medicine there are lots of anti-inflammatory drugs, and they are divided into steroid and non-steroid. And exactly, glucocorticoids, they are the prototypes of steroid anti-inflammatory drugs, because these are the steroid hormones. Steroids, they are produced from the cholesterol. That's why what is important to highlight that in both zones, lipid droplets are found inside the cell. Why? Because lipids are used as a precursor for those hormones, as they are steroid in nature. So which organelles should be more developed here? Let's remember cytology. Of course, smooth endoplasmic reticulum, and in mitochondria, here crests would have not lamellar structure, not plate-like structure, but instead tubular structure. And tubular crests, cresta, these are a signs of the steroid activity. So cells producing steroids, they have tubular cresta inside the mitochondria. Okay. So those two zones, they are actively stained with a special dyes like Sudan. Sudan, it stains lipids. So this zone is rich in lipid droplets. And what is interesting is that cells from the zona glomerulosa, they are converted in the zona fasciculata. So they are formed, zona fasciculata is formed by transdifferentiation of the zona glomerulosa. Okay. The last zone is zona reticularis, and it produces weak androgens, both in males and females. That's what is important. So in humans it is not very important, as in humans there are gonads and they produce steroids like male or female. But why we should remember about this zone? Because in case of tumors in women, the male sex signs might develop, like beard, like distribution of fat in the body and something like that. So zona reticularis produces weak androgens. So in case of imbalance of sexual hormones in women, this zone should be checked. Okay, then what about the medulla? Medulla derives from quite another source, and there are chromophenic cells. They are called so because they are stained with the chromium salts. And there are two types of them, epinephrine producing and norepinephrine. So different cells, they produce those two hormones. And let's investigate them in details. Besides, ganglionic cells are also present, but now we talk about chromophenic cells. Chromophenic cells, they are modified neurons, and they produce epinephrine or norepinephrine, which are used in nervous system as mediators. But here in the medulla, they are injected to the bloodstream, and they are used as a hormone, as a stress hormone, stimulating the heartbeat rate, regulating the activity of our sphincters, peristaltic distribution of the blood, and in physiology you'll investigate a lot about them. And what do I want to highlight? That epinephrine is also called adrenaline, norepinephrine, noradrenaline. The same meaning, because epinephros means the same as adren, around kidney means. So in different languages, different synonyms are used, so you should know both of them. Okay, what activates chromophenic cells? Chromophenic cells are activated by sympathetic nerve endings, so they are under the control of the nervous system. And they contain granules with norepinephrine or epinephrine. Different cells, they produce different hormones. So epinephrine producing cells, they have smaller granules. And except those two hormones, they contain chromogranin protein, and this protein is discharged to the blood or lymph together with epinephrine or norepinephrine, and it is detected in the bloodstream, and sometimes it is used as the marker of neuroendocrine tumors. Because chromogranin could be found not only in the chromophenic cells, but also in the parathyroid, in the pancreas, in many other neuroendocrine cells. The function is not very well understood, but it could be transformed into some statins like vasostatin, pancreastatin, and also it might inhibit, by the negative feedback, the activity of those chromophenic cells. And I want you to remember this because it is used in practical medicine as the marker of neuroendocrine cells. Okay, then norepinephrine could be converted into the epinephrine, and that's what is stimulated under the glucocorticoids. Okay, then the last information here is that ganglion cells also could be found, and these are like neurons with their nerve endings sent to the cortex they regulate the activity of the cortex, and also they regulate the diameter of blood vessels and the blood circulation in the adrenal gland. Okay. And several words about the blood supply. It is not important to remember like in the pituitary gland, because in pituitary it is extremely important. Here, just in general, I want to highlight that blood capillaries, they derive from the adrenal capsular arteries, and blood capillaries are formed either in the cortex or directly in the medulla. But from the first capillary network, blood descends to the medulla. That's why some glucocorticoids, they descend to the medulla, stimulating the conversion of norepinephrine to the epinephrine. Okay, so epinephrocytes would be more closely located to the cortex than norepinephrocytes. That's what I want to highlight. And then large central veins, they transport all those hormones from the adrenal to the circulatory system. And those veins, they are equipped with a well-developed smooth muscle cells. And contraction of those smooth muscle cells in the adrenal medulla, they stimulate like squeezing the sponge effect. They squeeze the adrenal gland and stimulating all those hormones to be transported to the bloodstream. Okay, and the most last slide, my congratulations if you're still here, and embryonic development of the adrenal gland. It has two sources. In general, medulla derives from the neural crest. As I told you, chromophenic cells, ganglion cells, these are neurons by their nature. And the cortex, it derives from the mesoderm, okay, in general, in brief. But if you want details, of course, you're curious, and fetal cortex is formed before the birth, and it represents the first wave of migration. So fetal cortex is formed from the mesoderm, which is located between the dorsal mesentery and the primitive gonads. So exactly from this region, intermediate mesoderm sends the first wave of cells, and they form the fetal cortex around the medulla. After that, the permanent cortex is formed by the second wave of migration from the mesothelium mesoderm of the dorsal posterior body wall. So from here, from the posterior body wall, mesoderm sends another wave. And the permanent cortex, it forms the zona glomerulosa predominantly. And first, just after the birth, the fetal cortex prevails, and only after that, permanent cortex develops with time. And before the birth, the production of steroid hormones, the hormones of adrenal cortex, is supported by the placenta, because adrenal cortex can't produce itself all the hormones. It needs placental support, so hormones are exchanged between them. Okay, so this is the last slide. Thank you for watching, and at the end, several questions from the USMLE database for the most curious and clever one. Okay, so medical student, very funny question, I decided to put it here. Medical student is learning histology. A photomicrograph is shown, and the question is, so which hormones are produced by the zone 5 predominant hormone, which is produced by the zone 5? So please be attentive, and if you've understood everything well, you will answer easily. Put your answers in the comments below and we'll discuss. And the next, much more complicated question, actually, if you are interested, maybe it is it has more clinical information, but I think that you'll cope if you think. So what is given in this task? 30-year-old woman, more or less young, yeah, relatively young. Her blood pressure was elevated, yeah, complaining her blood pressure was high, yeah. Then what about else? And blood pressure did not improve even after several trials of different antihypertensive medications. The question is, hypertension by which of the following structures is responsible for the patient's symptoms? So try to answer taking into account this information. Then the next question is about 47-year-old woman, and she's complaining of weight gain, so high body weight. Then also she feeling weakness, she's tired too quickly, her blood pressure is a little bit low, yeah, and thyroid gland is mildly enlarged. So which of the following antibodies is most likely to be found in the patient's blood? If you will listen attentively, you'll answer this question. So please put your answers in the comments. And the last one is 4-month-old girl, just brought to the pediatrician. She has a cough, difficulty swallowing, fever, seems like respiratory infection, and this is the 5th time her daughter has an upper respiratory infection. Along the 4 months after the birth, 5th time she has an infection. So we suspect the immunodeficiency, I guess, yeah. And also seizures, that's what you have to keep in mind. The oropharynx erythematose. And the question is, this infant's most likely underlying condition is associated with an abnormal development of which of the following embryonic structures? So try to connect this immunodeficiency, seizures, and embryonic structure. Put your answers in the comments below, and thank you for participating. Okay, and if you want more lectures on our subject, then please write down the topic below in the comments, and we'll consider each one. Thank you for the attention. Goodbye.